Hello, everyone, and welcome to Improve the Inclusion and Exclusion Criteria for Your Next Clinical Trial, presented by Roe. I'm Michael Gibney, and I'll be moderating this webinar. Our speaker today is Jack Modell, MD, Senior Medical Officer at Roe. You can read his full bio on the right side of your window. Just a few technical notes before we begin. If you have trouble reading a slide, please hold and drag the right corner of the slide window to enlarge. Please also disable your pop-up blocker to participate in the interactive parts of this presentation. We will follow the presentations by a Q&A session. Please submit your questions during or after the presentations using the Q&A box at the right side of your window. Okay, now let's begin. Jack, please go ahead.
Okay, well, thank you very much. Let me move up to our first slide here. I want to welcome all of you to this. I understand that we have participants from around the globe. So good morning, good evening, good afternoon, good night, wherever you may be. And I want to give a special shout out to my friends in the UK who are on today. So really appreciate this. You know, as much as I wish this were the most exciting topic in the world and could show you pictures of my trip to Italy last summer, there's a couple problems. One, I didn't go to Italy last summer. And two, while this may not be the most exciting topic in the world, it is certainly a very important one. Because obviously when we do clinical trials, as we'll talk about, it's the inclusion and exclusion criteria that really can make or break the success of a study, the interpretability of a study, regulatory acceptability of a study, not to mention just make day-to-day life easier, not having to interpret, explain, or worry about your inclusion-exclusion criteria. So moving ahead, as I just alluded to, the purpose of the presentation is to provide recommendations. Now, in a lot of cases, I'll be stating things that sound like hard fact and sound like absolute requirements. There are always exceptions, but in most cases, I'll try to give you my best understanding of just how important these things are. So I would take all of these at least as strong recommendations. But these are recommendations on how to formulate inclusion and exclusion criteria in your clinical trials to enhance the quality of subject selection and also to simplify and expedite the enrollment process. Now, in the lower right, you see the M&Ms. Now, I actually didn't pick the art for this presentation, but I will try to explain the art where I can. Interestingly, this picture, depending on the phase of the clinical trial, could go either way. For example, if you're doing an early phase trial, you might need to have a very homogeneous group of patients to test your new drug in, and that might be like starting from the mixed bowl and then parsing out exactly the individuals you want to study. Conversely, later phase trials, you may want to look widely across demographics, patient types, disease types, and put them all in your clinical trials for the broadest representation of what that drug can do. And I'll be more specific about that as well in just a bit. Another reason to really think about inclusion and exclusion criteria is to help minimize protocol violations, minimize amendments to protocol. Protocols, as you all know, is pretty frustrating once you're underway to have to change protocols because you have criteria that had unintended consequences or perhaps are difficult for folks to understand. So some general principles. Inclusion and exclusion criteria, as you know and as I mentioned, they actually define the population to which results of the clinical trial can be reasonably generalized. So obviously when you do a trial, you're not just looking at what that drug or what that product or device does in that specific subset of the population you picked, but you want to be able to generalize to a larger population. And depending on the phase of the trial and how you select your subjects, your study population may or may not be a good representation of the entire population with the disease or condition of concern. And that's fine as long as you keep in mind that you really want to pick the population so you can make the proper generalizations. Again, early phase, you might want just healthy controls, probably would want healthy controls, which obviously doesn't generalize to the population with the disease. And later on, you want to pick a much broader representation. Now, I will add that deviations from inclusion-exclusion criteria, which are sometimes called waivers, sometimes referred to by other terms, are simply not allowed, or at least per the FDA, and I know some other world regulatory agencies are strongly discouraged. And the reason is very simply because they potentially jeopardize your scientific integrity, regulatory acceptability, and subject safety. This comes up, for example, you may have a criterion, and I think we've all had this experience, where you might want to exclude people with hypertension. And you say subjects are excluded if they have a systolic blood pressure over 140. And then you get a call from a study site that says, well, this guy's blood pressure is 141. That's like 140. Can't I put the subject in? Well, of course, you have to scratch your head and wonder, why would the investigator call and tell you that? But given that he or she did, the answer is no. And some of you may be thinking, oh, come on, clinically, 140, 141, I agree that's insignificant. But if, when designing the study in the first place, you felt a 141 was appropriate, or a 143, or a 145, the cutoff should not have been 140. So all of your parameters around your inclusion-exclusion criteria, you have to really mean it. There can be no deviations from it because, as the FDA has pointed out, once you start to deviate, I think the words of the FDA, we don't really know the population that you're studying. You say on inclusion-exclusion criteria that it's a certain population, but then you've made all these little deviations, and in the end, we don't know quite how to interpret this population. And actually a story that comes to mind, probably shouldn't, but back when I was in about third grade, I had a very forgiving art teacher who gave us an assignment to basically color within the lines. Well, like I imagine a lot of people on this call, if you're told to stay within the lines, what's the first thing you have to do? Well, I colored all over the place. And she came up to me, and while she could have been critical, she said, well, that's really very nice, but what is it? And I probably made something up, but that's exactly the point. Once you start going outside the lines, regulators, yourself, others lose track of exactly what it is, and you don't want to do that. So no protocol deviations, or waivers, I should say. Protocol deviations happen, but not waivers to inclusion and exclusion criteria. So some general principles, and again, people on the call know this because you're already in the field, but I go through these basically to help as you go through protocols to really think about the purpose of these criteria, which I think makes it easier to write them more cleanly and more succinctly. Inclusion criteria should contain only those items that define the target population intended for study. So sometimes you read through and you see things in inclusion criteria that talk about which patients can't get in, or things that don't really define the target population, and those criteria need to go elsewhere. So this is just the items that define the target population. And within the limits of the known safety coverage, these criteria should generally allow you to include the broadest representation of the population for intended use of the product, but usually not beyond. And what I mean by that, obviously, is you don't want to go into a very large study with subjects with illnesses or nature of disease that you don't know if the drug is safe for. On the other hand, if you limit it too much, and I'll give you an example, you may lose a lot of important information about your drug. So a good example was in some of the early hypertensive trials, for whatever reason, probably not a good one, a lot of the inclusion and exclusion criteria specified Caucasian population. Now, a lot of times it just happened that way because, as you all know, minorities were under-recruited and underrepresented for the longest time, and fortunately that's largely been corrected with increased awareness. But some of the early studies actually specified just Caucasians, not to be discriminatory, but to say, well, this is what we tested in phase 1. We want to be careful. We don't know how non-Caucasian populations might react. It then took another 20 years to figure out that some of these hypertensives didn't work as well in black patients, for example, or that certain antihypertensives did work better in the black population. So you don't want to unintentionally over-exclude subjects at all. So think as broadly as you can as long as it's safe for your clinical trials. And when you write these criteria, for each and every inclusion criterion written, you need to sit down and ask yourself why that criterion exactly as written is necessary to define the desired study population. And in doing that, and this is good to do with a team and not just one or two people, because very often people will, when you do that, say, well, wait a minute, this duplicates something above, or this doesn't quite define the study population that we want, or that there's just a simpler or clearer way of writing it. Exclusion criteria shape the included population by defining which subsets need to be excluded for other reasons such as safety. So, if you're thinking, I think in terms of like an artist, the inclusion criteria is the material that the artist is going to work with. The lump of clay, for example, or plaster or whatever. Your exclusion criteria then shape that by taking off those parts that, the analogy breaks down, but taking off those parts that don't give you exactly the kind of outcome, exactly the population, the shape of the population that you need to test the product. And like for every inclusion criterion written, for the exclusion criteria, you now need to go through and ask why that, exactly as written, is necessary not to include subjects, but in this case, think about it as preventing subjects who in some cases may otherwise be qualified from participating in the study. And very often there's good reason. I'll give you some examples later on where, unfortunately, very often there's not good reason. We sort of arbitrarily say we don't want somebody over 65, we don't want somebody over a certain blood pressure, under a certain age, a lot of different things that we're just so used to putting in protocols that we don't really think about whether we really need to do that. Okay. Also, as I go on, I can't see whether you're asking questions or not, but as the moderator said at the beginning, do feel free throughout to ask questions as they come up. I may not be able to get to them until the end of the presentation, but I would be very interested in questions you have or feedback. I'll be happy to address if there's time. So looking at general principles, these criteria need to be written as clearly and unambiguously as possible. In a way that's obvious, but again, for those with a lot of experience, we've seen plenty of cases where it's not written that way. For example, I saw a recent protocol where exclusion criteria said steroid use within the preceding 3 months. That caused a lot of problems. Did they mean corticosteroids? Actually, they did, but steroid usage could also include mineralocorticoids. So the sponsor got that straightened out, that they meant corticosteroids, but then came the question, well, did you mean parenteral? Did you mean oral? Did you mean topical? And originally they said all steroids, but it turned out, if you really think through this, for most clinical studies, if somebody puts a little bit of hydrocortisone cream, and I'm not talking about a dermatology study now, but a little hydrocortisone or topical steroid cream on small surface areas of the skin, it's not going to much affect anything, any of your outcomes. And yes, I know there are exceptions, but in general, no. So again, you want to think through and be very specific exactly, what are you trying to exclude and what are the limits of exclusion? Is it any use of something? Is it use extending beyond a certain parameter? And this is almost a repeat of what I've said, but there needs to be considerable forethought into the necessity, desirability, and potential unintended consequences of each criterion and any associated parameters. Another example that comes to mind, this was a study actually I looked at probably 3 or 4 years ago, where it was a drug for sexual dysfunction in men, and the inclusion criteria contained a criterion that said that they wanted men in, I think it was monogamous sexual relationships with women. And whoever wrote it, I'm not quite sure what they were thinking, but just thought, well, that maybe to them was the norm, and that's what they wrote. When I got a hold of the protocol, I said, wait a minute. First of all, does it have to be monogamous? And second of all, why strictly limited to heterosexual relationships? And the answers I got, frankly, were not very satisfying, and eventually that criterion was broadened. But, that would be an example where left unchecked, and I think in today's day and age, we probably would catch something like that. 10 years ago, not so much. But left unchecked would limit enrollment because you're excluding people who, are perfectly acceptable to be in the study. And you're limiting the generalizability of the study results because that product may be useful far beyond men who are in, single relationships with women. So again, thinking through each criterion and asking yourself, is there anything unintended that could happen and do we really need it? Going along with this, care should always be taken to write the fewest number of inclusion and exclusion criteria sufficient to describe the population being studied. As I think you've seen in a lot of cases, in life in general, but certainly in science and protocol, less is often better. The more you have, the more there is to conflict internally, and the more there is for people to stumble over. And as you know, if it's there, somebody's going to stumble over it. So be sure you look through and make sure that you don't have duplicate, overlapping, and potentially contradictory criteria. And what you see in green really is a summary. I'm not going to read through it, but again, look through it really carefully. Is this everything you need? And be sure that it isn't any more than you need. Now, one place where we really get into a lot of trouble is, and it looks like a copier has, at least on what I'm looking at, blocked off the words, but what that says is inclusion-exclusion criteria should not be copied indiscriminately from one protocol to the next. Criteria should be selected anew, I think I just wanted to use the word anew, for each protocol, though previously written criteria can be used for reference. Many, many times I think we've all seen criteria copied from one protocol to another, and often it just doesn't make sense to do so. And why do we do this? Well, sometimes I hear stuff like, well, the last study worked, so don't change anything. Well, the last study could have worked for a lot of reasons, and they could have worked in spite of the criteria that you had. So obviously, if you know something really helped, you don't need to change that. But you really do need to consider protocol anew for each protocol. Conversely, I've also seen, well, the last study didn't work, so we have to change everything. And again, there's lots of reasons why studies don't work, but be careful about that. Even in duplicate studies that are done, subsequently. So if you have one study done in 2012 and your next study is being done this year or next, things change. You've learned new things, the environment changes, regulations change, and you really need to ask yourself if changes are needed. And looks like we've got a slide problem here, but what this says is this applies to labs and clinical exams. And what's written there says, I have no idea how to interpret it or why it's there. Oh wait, it was in the last protocol we did. So this goes beyond inclusion-exclusion criteria, but it is a pet peeve of mine. So often, laboratories and exams are ordered, and the study team and the investigators don't really know why it's there or how to interpret it. And we had a recent case, without giving away too much information, of some clinical labs that were ordered on subjects, copied from the last protocol, that gave information that just wasn't relevant to that population. And lo and behold, a bunch of these labs started coming back abnormal. Turned out to be the lab's fault. Well, the lab didn't think so, but we think so. So it was lab error, but now we're in the position of deciding what to do with abnormal labs that probably mean nothing in a population where it wasn't needed, and explaining, well, don't worry about it, we really didn't need to get them anyway. So be especially careful in your criteria and laboratories that you're ordering, making sure that you really need everything, that you're not leaving out things that you do need that you wish you had, and that you're going to know how to interpret high results, low results, and normal results. And, if the answer is, well, we don't really care if it's twice normal or half normal, there's a good chance it doesn't need actually to be in there. And there's sort of a false sense, too, that regulators require everything. They don't. Regulators require you to do a thorough safety assessment, obviously, but Any regulator I've talked to says that that does not mean ordering as many tests as you can or doing as many evaluations as you can. And as long as there's a good reason for ordering what you do and for not ordering what you don't, they'll be happy with it, which makes sense because that's exactly the case in clinical practice. You also might want to think about inclusion-exclusion criteria in terms of where you want to draw the line between sensitivity, meaning picking up something that's there, and a false positive exclusion rate. Recognizing that there's no perfect answer and there's actually a reciprocal relationship between the two. And what I mean by this is, let's say we're doing a depression study and we only want patients who have major depression in the study. And let's say we have a scale, and this doesn't actually fit any particular depression scale, but let's say that if you have a 10, you're very mildly depressed, and a 20 is moderately depressed, and a 30 is severely depressed. And you say, you know what, we don't want to miss any depressed subjects, so let's take everybody that's a 10 or above. So that would be like picking something to the right side of that diagram where the sensitivity is high. Pretty much anyone who's depressed will have a 10 or above, but an awful lot of people who are just having a bad day are going to have a 10 or above. So you'll have a high false positive rate. On the other hand, if you say, well, you know what, we don't want people with normal mood variation or people who are mildly depressed. We really only want the sickest of the sick, and we're going to make it a 30. Well, that's like pushing it way over to the left, where you're going to miss 80% of your depressed subjects, but you're not going to have any false positives because someone who's just having a bad day is not going to hit a 30. At least I hope not. And if they do, they probably ought to see someone because it's probably a major depressive episode. So actually this is just another example which I'll take you through quickly. If you want no hypertensive subjects, well, exclude everybody with a systolic greater than 120. Well, would you do that? No, probably not. That gives you very high sensitivity for detecting hypertension because you're not going to miss patients who are truly hypertensive. On the other hand, you're going to inappropriately exclude a very high percentage of subjects who don't have hypertension. So you could go the other way. Just exclude all subjects with a systolic pressure of greater than 180. Well, that will give you very low sensitivity for detecting hypertension because you're going to miss most of the subjects who have it. On the other hand, there won't be any non-hypertensive subjects who are excluded. Now, as I mentioned earlier, I didn't put these pictures in here, and I have to tell you, I'm looking at this kid who, again, reminds me of me in my art class, and wondering what this has to do with hypertension. So if any of you have any funny captions for this or any explanation of how this child is related to hypertension, send it along, and I'll be happy to read it at the end. Okay, so clearly, as I've described with hypertension and depression, no matter where you set a criterion, there's going to be a trade-off between subjects who are unnecessarily excluded and those who probably should be excluded but don't meet the specified criterion. So the study team just has to decide what the best trade-off is. And a lot of times where I've seen study teams really go awry in this area is with black and white thinking, saying, we have to include such patients, we must not include such patients, and where do we put it so we get the black or the white? That's rarely going to happen that way, and it's much more about, where do you want that trade-off line to be? So try to avoid looking at these things in categorical terms. All right. Your criterion and indeed your entire protocol are really only as good as your investigator education. And as I'm sure those of you who have worked with investigators know, this is not necessarily an easy task, but it's a very important task. So study teams do need to be diligent in reviewing the criteria with investigators to be certain that the protocol specified details are completely understood and that they understand, as we talked about earlier, that these criteria are not to be violated. And I also find that it's very, very helpful, especially at investigator meetings, to explain to the investigators the rationale for each and every inclusion-exclusion criteria. Why this may seem a little bit painstaking, there's a couple good reasons to do it. One is if they understand the rationale, they're much more likely to adhere to it and not violate it, deviate it, or get around it in other ways. And as you know, investigators find ways. But if they get it, they really get the broader picture, which is helpful. The other reason to explain the rationale is it's really interesting. You can sit around a table with the study team and have great rationales for a lot of things that you do. And it's sort of like the minute you send an email, that's when you realize, oops, it wasn't as perfect as I thought. The minute you start explaining to investigators, either you or the investigators are going to say, wait a minute, no, that doesn't quite achieve what you're trying to do. And so that's very helpful at that point to kind of hone down the inclusion-exclusion criteria and make sure that you're getting exactly what you want. Likewise, not just in investigator meetings, don't be shy or hesitant in the protocol itself to include explanatory footnotes or explanations about why certain criteria are as they are, especially if they are a little bit difficult, complex, or you think investigators aren't likely to understand or accept it. The criteria do differ across phases of development. I think clearly enough that early in development, when you don't have a lot of safety information, generally we use more conservative criteria to really ensure that the patients are safe, because we don't really know how the drug is going to work, say in a less selected population that might have, shall we say, normal wear and tear diseases, obesity, hypertension, diabetes. So that's where we tend to get the healthy controls. But once the safety profile is established, then we can start using less restrictive criteria so that you get more of a real-world experience in the type of patients that are actually likely to take the drug. And then it also helps to define the appropriate labeling for this target population. For example, going back to the hypertensive trials, had they included all races, especially Caucasian and Black, in the early trials, they would have seen right off the bat that the drugs work better in some populations than others and have been able to explain that in the label. Now, there was a time, I hope it was in the past, when a lot of pharmaceutical companies might have known or suspected that a product might not work as well in a particular population, and they intentionally excluded them, saying, well, we just wanted a particular population because that's where it's going to have the best effect. If that's what the label is going to say, and that's the limit of your intended population, that's okay, although I would ask, I would still suggest you test it in the other populations. But what you absolutely don't want to do is intentionally exclude populations or patient subtypes where you don't think the drug is likely to be as safe or efficacious, yet you're hoping your label will include them anyway. It's not appropriate, it's not ethical, and that shouldn't be done. Well, moving on. You also, in writing the criteria, need to consider the target population. And what I mean by that is healthy subjects, for example, versus subjects with diseases may have different values, different parameters that might be acceptable or not, or might even define pathology or not. So for example, what I mean is that let's say you have an athlete or a runner with a resting heart rate of 45. That's fine, unless you happen to be testing a drug that slows heart rate even further. Maybe that's not the ideal subject, but for most trials, there's nothing wrong with that. Yet I have seen an awful lot of trials say things like, the subject has to have no EKG abnormalities. And actually very few of us have no EKG abnormalities. Or subject has to be in normal sinus rhythm. Well, what you see here is a sinus rhythm. It's basically a normal rhythm. Technically, it's a sinus bradycardia, which is not a normal sinus rhythm. But the point is, you don't want to have to get calls saying, can I include this subject? Because if the subject is healthy and that's why his or her rate is 45, there shouldn't be any problem. Conversely, if you're doing a study in cardiac patients, a sinus bradycardia often signals a significant underlying pathology or drug effect. And so that's why that the team and the protocol have to differentiate between normal physiology that may produce a technical abnormality, if you will, and a truly pathologic condition. And I'll get to some examples of how you can clarify that by using very specific criteria. Okay, looking at disease area, this actually is a repeat of what I just said. You need to adapt the values, your ranges, to the population under study. This is just another example here. You know, you may, in a lot of studies, not want anemic patients, patients with heart rates of 110, patients with hypertension. But if you're studying a population where that's really common or part of the disease, you need to be sure that your criteria reflect that appropriately. And I have seen a lot of people get hung up studying, say, chronic diseases, saying, well, we really don't want anemic patients, so anybody who has a hematocrit less than 33 can't get in the study, forgetting that hematocrits of less than 33 are pretty common in chronic diseases. Likewise, I saw a study for generalized anxiety where they said, well, we don't want people who have really fast heart rates. Now, if you're testing a stimulant, which you probably wouldn't test in generalized anxiety, I could understand that perhaps, but by this particular study said anybody with a resting heart rate over 100 can't get in the study. They were dealing with anxious patients who, when they came to the investigator, were even more anxious, and that excluded unnecessarily about half the subjects. So if your goal is to exclude a specific medical problem, for example, a serious cardiac arrhythmia, then that's what the criteria should say, to the best that you can, not just any cardiac abnormality. And not, like I said in the example that I showed, if you say something like cardiac arrhythmia, to a lot of people, well, that sounds terrible, cardiac arrhythmia. Well, it's not, because there's an awful lot of them, and if we've got 200 or 300 people on this call, I would say, not to scare you, but probably at least 40 of you have some kind of arrhythmia right now. It could be a tachycardia, bradycardia, PVCs, PACs, yet you all may be appropriate to be used as healthy test subjects. So please be very specific about what you're trying to exclude and to know why. We also need to be careful with so-called demographic or standard inclusion-exclusion criteria. Very often we see studies that say ages 18 to 65. Well, there's a reason in a lot of cases for the 18. But why the 65? I mean, does something happen at 66? I hope not, because I'm less than 10 years away from that. So, be careful setting things like age and ask yourself for what reasons, because we see this very often. Now, BMI actually is an interesting one, because BMI, while I'm telling you here, don't have arbitrary criteria, it's very interesting that a lot of times studies will say, well, you know what, we don't want hypertensive subjects and we don't want patients likely to be sick or have comorbidities, but geez, there are an awful lot of overweight people, especially in the U.S., so let's allow a BMI up to 35 or 40, not realizing that obesity itself is a disease. And by the time you start getting BMIs of 30 to 35, you tend to get into populations that have higher comorbidities or a greater risk of potentially serious outcomes, not just from your drug, but unfortunately, from the obesity in general. So in setting BMI, find the right range where, you're not going too low that you're unnecessarily excluding healthy subjects, which may be around 30. Although, just to give you an example, 30 would be somebody who's 5'10" and weighs 210 pounds. A BMI of 35 is someone who's 5'10" and weighs 245, and I've even seen BMIs as high as 40, which would be a 5'10" who weighs 275 pounds. So I would try to keep BMI at a point that is commensurate with your other degree of risk that you're tolerating. Don't go too high if you're concerned about comorbidities, but obviously don't go too low unless there's a clinical reason to do it as well, or you may over-exclude. So again, like I've said, rather than saying a cutoff of 65, if a healthy 80-year-old is acceptable and a 50-year-old in poor condition, isn't acceptable, then age shouldn't be the limiting factor, but it should be specific diseases or concerns. So you could write something more general, like no major illness or debility that in the investigator's opinion is likely to jeopardize subject safety or interfere with the subject's ability to comply with the study requirements. On the surface, that sounds great. Sometimes it is, especially for small studies, if you really know the investigators well. The problem is, there's a but there at the bottom. If you're going to use more flexible criteria, you have to have very strong assurance that these decisions can and will be properly made, and that variability or errors in investigator judgment are not likely to skew the intended population or jeopardize patient safety. Now, for those who perhaps haven't been investigators or haven't monitored investigators, a lot of people think, well, doctors know better. Doctors know if somebody's too sick. You can trust the investigators. Gosh, if you can't trust the investigators, why are we even doing the study? I don't know. That's a good question. But let me tell you that for good reasons and not so good reasons, investigators often have a lot of difficulty with flexible criteria, often taking liberties that they shouldn't. Why? Well, sometimes they don't know better. Sometimes it's because the patient really wants, or the subject really wants to be in the trial and the investigator is trying to help him or her. Sometimes the investigator feels incentivized to get subjects in. And I'll give you a quick example. A depression study several years ago that I was doing required a certain level of depression to be in the study. And I actually told one of my cousins in Chicago about this. And he said, because I knew he had depression, and he went to one of our investigative sites. And he called me and he said, Jack, it's an interesting process, this getting screened, because I told the investigator the severity of my depression, and he did a little test, and I came out to be a 16. And he said, well, you need an 18 to get into this trial. Are you sure you weren't depressed a little more than, whatever it was that you said? So investigators will often coach subjects, as you know, or use their own interpretations to get people in. So be as specific as you can. So as I say, the use of these criteria does require, and this was supposed to be a build actually, I guess we lost that, but I'm going to go through these. You have to be sure the decisions can and will be properly made. The first one, these are all actual quotes that I've heard. I'm a psychiatrist. How was I to know that the subject had pneumonia? Your mother knows if you have pneumonia, and psychiatrists know all about mothers. They should know if you have pneumonia. This was a case where the criteria basically said good general health, no major medical diseases, and the psychiatrist, I don't know how he missed the pneumonia. This next one was actually a gastroenterologist. Well, I knew his liver enzymes were elevated, and in this case it was about 2.5 times normal, but I've seen much worse. Of course he has. He's a gastroenterologist. And the subject really wanted to be in the study, so they put the subject in. Next one. The rating forms didn't show it, but in my clinical opinion, the subject had it, so I put him in the study. Again, if you're leaving it to investigator opinion, you're going to get this sort of thing. Another one, I don't remember the specialty, but the protocol said no current major medical illnesses, and her colon resection for cancer was 7 months ago. Should have been clear. The last one was from a surgeon who said beta thalassemia is a major illness. Actually, in fairness, beta thalassemia can be minor. In this case, it wasn't. So again, leaving things to investigators, it's pretty risky. You can't just say, well, they're doctors, they should know better, because we all know the old joke about, trust me, I'm a doctor. It doesn't engender trust. And no offense, I'm a physician too. Most of them do great jobs, but this is science we're doing here, so you don't want to leave too much guesswork. So it's also permissible, and to be honest, this is what I favor, just because of the experiences I've had, to use fixed, or some people would say rigid, exclusion criteria. For example, you don't want anyone with a sodium of less than 130 or greater than 150, systolic blood pressure less than 60, it's a little low actually, or greater than 160. If there's clinical justification for why it's desirable to include exclude subjects, and that these values are intended to be unequivocal. The advantage is that it sets absolute standards for subject participation that are not open for opinion or debate. And I'll show you an example of this in just a minute. The disadvantage, if the criteria are too stringent, then they could unnecessarily exclude some subjects. So for example, I don't want anyone with a blood pressure of greater than 140. Well, like I said, if a 141 is okay, make it a 150 or a 160. But if you really think through them carefully, they can really be helpful. This slide, it's hard for me to see. I hope it's not as hard as I think it is for you guys to see. But this is an example of fixed exclusion criteria that we use from a late phase antidepressant program. And I'm not going to go through all of this, but in the upper right, we basically showed a table that said if the subject has any of these lab results, the subject is not to be included. So for example, I'm looking at the glucose, which I can read. It's about a little more than halfway down the chart, less than 60 or greater than 160. It was so easy. We actually got no or almost no calls on inclusion-exclusion criteria in this protocol. If we set a 160 and the glucose was 161, end of story, the subject was not in the study and we didn't have to get called about it. I was talking earlier about EKGs. If you look under the table, I think it's criterion number 23. Subject has EKG, or it's going to be hard for me to read this, or clinical evidence of ventricular hypertrophy, intraventricular conduction defects, I think cor pulmonale, myocardial strain, ischemia, infarct, et cetera, second or third degree AV block. We were very specific about what couldn't get in. Now, some people say, well, you'll over-exclude subjects. There probably weren't more than 3 or 4 subjects that were excluded because of this. But the investigator said, it made it very easy. Because when you get EKG readouts, very often there's nonspecific ST T-wave changes, sinus bradycardia, unspecified abnormality, 2 PVCs per minute, and they didn't know what to do with it. With something this clear, they didn't have to know what to do with it. They knew what was in and what was out. But obviously, if there was something that they were concerned about, they could still make their own judgment to exclude. The other thing that I commonly include, and this is number 21 now on the left side, you don't necessarily have to do it exactly this way, but drug tests and alcohol tests are really important because, we really want, and just to be clear, I'm not making moral or ethical judgments about drug or alcohol use. I'm actually pretty liberal about these things, but when it comes to science and studies, you need to know what's in your patient, and you need to have pretty reliable patients. And on the whole, especially subjects that use illegal drugs, and yes, this is a generalization, but they're less likely to be open and honest with you about certainly drug use and maybe some other medical things than those who don't. Now, granted, anyone can be honest, anyone can be dishonest, but since your inclusion-exclusion criteria, all of them really deal with probabilities, and you're trying to do the best you can to get the population that you want, eliminating subjects who are illegally using drugs is generally a pretty good idea. And do not allow repeats. And the reason is, urine test repeats. And the reason is, yes, you hear, well, I ate a poppy seed bagel. The chance that the subject is being dishonest with you about why their urine is positive for opiates or whatever you're checking is far greater than the chance that the subject ate enough poppy seeds, with today's test needs to be a lot, that they just happen to turn up positive. And they just happen to eat them 6 hours before coming in. So generally, we don't allow repeats. You can see we're very specific here with the blood alcohol level. You could just say no blood alcohol level. I remember for this particular study, I think the argument was, well, we were doing it in Europe and people there drink a lot of wine, as if to say they don't in the U.S. and elsewhere they do. And we didn't want to exclude someone who had a glass of wine several hours before. Well, actually, what we ended up doing, and it worked very well, is telling subjects not to have any alcohol within at least 8 hours of the screening visit, because frankly, if they couldn't adhere to that, we were concerned about overall adherence. And their level had to be 0 or near 0. And just in case some are wondering, well, what about this 0.015? Frankly, I would have rather it been 0, but to have a 0.015 8 hours after drinking, you would have to have over about a 0.12, so well over the legal limit, and about a 6-pack's worth 8 hours before. And anybody who's drinking that much, well, let me qualify. Many people who are drinking that much have problems that you don't necessarily want to have in the clinical study, unless, of course, that's what you're studying. Okay, so moving on a bit, obviously some studies go on a very long time. And in those cases, you should think about, rather than using specific wording for some things, if you're using a marketed drug, you can use wording from the product label, from the product information, such as, the use needs to be within prescribing information. That way, you don't have to keep changing the protocol every time the label changes. So this may not come up that long, but for long duration studies of marketed products, it's often a good idea. And then obviously include the product label with the protocol and make sure the investigators understand it. Some regulatory and ethical considerations. In later stage development especially, your inclusion-exclusion criteria may have regulatory implications, so certainly consider that. But in all cases, please, always, the goal is to optimize the benefit risk for patients, not to optimize the number of sales, not to optimize the broadness of the label, unless you can do that without also ensuring safety and efficacy for patients. So safety and efficacy is foremost, but otherwise, as we said, broad criteria for inclusion-exclusion, as long as it's safe to do it, and as long as it gives you the population where you're trying to generalize the results from. Something that, honestly, I didn't even know happened, maybe I should have, until fairly recently, is I've seen sponsors tell investigators, and I frankly have seen investigators who do their own research, who will groom, if you will, subjects. So they're not subjects. So they'll have patients of their own, so clinic patients. And they know there's a study coming up in 3 months or 6 months, or maybe it's already there, and they're thinking, well, you don't quite meet inclusion-exclusion criteria today, but if we change this medication or if you do this or that, in a few months, we could put you in the study. That is not to be done. It's unethical. It's prohibited for 2 reasons, probably more as I think through this. But one is until they've signed the consent form, they're not a research subject. And so you don't do things to a subject or potential subject that is specifically for the research protocol until they've signed the consent. The second thing is, for any clinic patient, what you do clinically has to be what is right for that patient clinically. So if you have a patient who's on a medication, you're obviously prescribing it because you think it's the right thing to do. So you don't stop it so they can get into the study. Now you may say, well, what if they don't need it? Well, if they don't need it, then I would say if that's really the reason, then the clinician should do whatever he or she needs to do with that patient's medication to optimize that patient's health. And then if the subject or the patient qualifies later on, that's fine. But it should not be done primarily for the purpose of getting the subject into the study. A couple operational issues. I think we've probably all experienced this. Subjects get randomized before you have all the information back relevant to inclusion-exclusion criteria, such as laboratories. So one thing to do is when you're writing the criteria, writing the protocol, allow enough time to get back what you need before you put the subjects in. And sometimes I've heard people say, well, we really don't need all the labs back. I mean, it would be nice, but if this lab or that lab doesn't come back, it's no big deal. I would say if it's no big deal, then why is it in the inclusion-exclusion criteria to begin with? But once it's there, you can certainly build in check steps to your programming so that randomization simply can't occur until all the criteria have been entered and noted to be consistent with your protocol specifications. So we don't have too much time, and this actually was intended to be a take-home slide. So let me just explain the goal here, and we may have a few minutes for people just to write some things down. But let's say that, there we go. that you're writing inclusion-exclusion criteria for a depression study. And for the purpose of what we're going to do right now, ignore the 4 and ignore the 6. Just be thinking about you want to have inclusion-exclusion criteria that are as important, as precise as possible. So let's say we're testing a drug for depression, call it 150 per arm. It's been already tested in a healthy volunteer study, no major problems. And by the way, when you get information that says a drug is well tolerated, many of you know this already, the devil is in the details. Well tolerated can mean a lot of things. Please read carefully what that tolerability was, because sometimes well tolerated could mean no problems at all, and sometimes it could mean no serious problems, but there's a lot of room in between. And let's just say that in the phase 1 studies, prolonged QTc interval is observed in 4 out of 30 subjects on drug compared to 2 out of 30 on placebo. You probably wouldn't run stats on that, but if you did with a Fisher's exact, you'd get a P of around 0.7. But you didn't see any dysrhythmias. So just take a minute and just send in a couple of inclusion-exclusion criteria. If you can't think of a couple, it could be one. We have enough people out there that if you each send me an inclusion criterion or an exclusion criterion that you think would be important to have in there, go ahead and take a minute and do that now. I'll stop talking for just a minute, if I can. Might be hard. And we'll get those in, and then when we get to the end, be able to discuss them. And I don't mind humor, so if you have anything funny to say, if it's politically correct enough to read, I'll probably read it. Questions coming through? Okay, steal this nice person's laptop here. Question that has come through, or a comment: it is important for people developing protocols to incorporate this thinking while writing inclusion-exclusion criteria, especially if the trial is a pivotal trial. I guess that's a statement. Yes, it is. Very important, yes, especially for pivotal trials, because those are the ones that potentially can affect your labeling the greatest. And again, you don't want to underrepresent populations that could potentially be helped by the drug, or in which you think the drug may not be so well tolerated. Obviously, you don't want to hurt anybody, but you don't want to exclude populations just because you think they may have slightly more adverse effects and you don't want the FDA to know about that. That's generally not a good strategy. Another question? Since we're near the end, if you want to go ahead, if you don't have much to say about the depression protocol, you can certainly think about that later. Feel free to send in other questions as well. Does the inclusion and exclusion criteria, are they country specific, just as you mentioned, for it to be population specific? Well, I can't speak for every country. I've done a lot of global studies, and in general what I've said would apply globally. Obviously, if you know of exceptions or your regulators, have asked for something specific, that needs to be included. But pretty much for everything I've covered, we're talking more science here than what's a regulatory requirement. Although generally most regulators, there are some exceptions, but generally they go with the science. So I would say in general these are broadly applicable, although there may be some minor exceptions. Let's see. Not quite sure how to interpret that one. There's a question about, I guess I'll ask the question, maybe I'll think of the answer while I'm asking, are there mentionable differences, maybe as opposed to unmentionable ones, between treatments that are long-term, weeks to lifelong, versus short-term treatments, a few days? Not quite sure what that means. Obviously, yes. Longer-term trials, when writing the protocol, the longer it is, the harder it's going to be for subjects to comply. The more adverse events, the more problems can crop up. But in terms of inclusion-exclusion criteria, I think these principles apply regardless. Let's see. I can flip through these. Do you exclude subjects currently enrolled in another trial? The answer to that is almost always yes. There may be some exceptions, but you don't want subjects getting 2 experimental treatments at one time. You don't necessarily know how they're going to react, and granted, one could be cream, on your foot for a fungus, and the other could be something on your hand, but in general, and I know in some cases even regulators specifically say you shouldn't. Let's see, how can I scroll through these? There's a QT question. All right. Okay, inclusion. QT interval within normal range. Document what is normal. Yeah, that's actually a good point. Okay, we're showing the questions now, not the slides, but that's okay. There was a little bit of a trick in that discussion, and maybe this person picked up on that. I mentioned the prolonged QT interval. You saw it in 4 out of 30 on drug compared to 2 out of 30 on placebo, but no dysrhythmias. That's not very reassuring. Frankly, that ratio, if you had, I think it's around 150 or 180 patients, and you have the 2-to-1 ratio, that would be statistically significant. So it doesn't mean it's significant there, but it means that even though the p-value is insignificant, it does not mean that there's no QT issue. So actually the QT in this case does bear further exploration. And frankly, by the time you're going into late phase 2 testing, you ought to have more QT information. But if you didn't, yes, I would be concerned about putting this into subjects at risk for prolonged QT. So that's very good. What are your thoughts on exclusion criteria, such as study site or sponsor employee or relative of an employee who's directly involved in the study? That's a good question. In general, it is frowned upon because to include people who are at the site, friends, colleagues, because it could be considered that there's undue coercion. Although I can tell you back in the 80s and 90s when I was doing my own research, that was called convenient. And we did a lot of studies. In fact, I did one in which I was a control, I think. And that was considered convenient. It was fine. And the view was, well, geez, we're all consenting adults. There's no reason that we can't. I actually don't know if there are laws about it now. I do definitely know that there are guidances and guidelines that say that people who are closely related to investigators or the study site should not participate because there are potential conflicts of interest. So my response to that is, if there's a reason to say that, and I have a feeling there is, I don't know the latest on it, but I don't think that's unreasonable.
Jack, I'll jump in now. That's about all the time we have. Okay. We had a lot of great questions today and couldn't get to them all, but we will be getting back to everyone who submitted personally after the webinar. Thank you for attending this Fierce Live webinar and submitting so many great questions. I'd like to thank our speaker for participating, Jack, and Ro for presenting today's webinar. This webinar has been recorded. You will be able to access the recording within 24 hours on the same page you used to register for this event. Thank you again for joining, and we look forward to seeing you at future events.